So this is for DNA replication. I'm going to be speaking about the basic form of DNA replication, and then I'm going to be speaking about the fancy form or the more advanced form. But just to get our orientation, let's go ahead and speak about the DNA rather quickly. The DNA is double-stranded. The top one will be the number sign, and the bottom one will be the asterisk or the star one, the blue. So when we speak about replication, these strands are now not just ordinary strands, now they're considered parent strands or old strands. So the top one is an old strand, and the bottom one, the star one, is an old strand. Once the pound or the pound DNA strand leaves and the asterisk DNA strand leaves, they become you could say their copies, but I guess you could say of each other, but they become this strand here, the old strand. Remember the top one, um, gets a freshly made new strand. I guess you could say here in the bottom, and it's basically it is called the new strand. Another term for it you may know is it's called the complementary strand. This old strand, remember the one on the top, it is the old strand, but we could also another term for it is the parent strand. Okay, so now speaking about the basic form of DNA replication. So let's go ahead and move to this diagram here. So as we mentioned, the old strand is right here. Remember, let's say the number sign. And then the other old strand is right here. Let's say this is the asterisk. The first thing that happens is that DNA unwinds at its origin, and its origin just basically means right here in this section. And then the second part, supercoiling, is basically a side effect if you want to unwind it. You could think of getting like a string that you found on, let's say, your piece of cloth. Once you keep pulling it, there's going to be a moment where it stops because of the supercoiling. It'll be so coiled that you won't be able to pull the string anymore. So the third step, and then we'll go into more detail in a second. The third step is that the new strand at the top is made continuously. So you could think of it like a freeway. It's made nonstop, continuously. The fourth step is that the new strand at the bottom is made discontinuously. You kind of think of it like if you go into your home, but instead of taking the freeway, you take the streets which have intersections. So you have stops and potholes and so forth. So let's continue on. So a short sequence of RNA nucleotides are added to this strand here. And you could just think of it like RNA nucleotides. The first thing that comes up is uracil, right? So let's just say that uracil is added, a triplet of uracils are added to the new strand here. Now that that's added, then an elongated sequence of DNA nucleotides replaces the uracil with adenine. The discontinuous fragments, because again it's made in pieces, right? These continuous fragments are then glued to make DNA. So that's the basic form of DNA replication. Now let's go into more detail. So again, we're saying the same exact 5 steps, but now we're going to add more details. So now Helicase unwinds DNA at its origin of replication. H is for Helicase. Topoisomerase relieves the supercoiling. The leading strand, you can tell that it's all straight lines with no gaps. This is the leading strand, which is the new strand. is synthesized continuously by DNA polymerase. So again, nonstop. The lagging strand, you can see the breaks here, which is the new complementary strand, is synthesized discontinuously or in parts. Primase synthesizes a series of short RNA primers. So the primase will be, you kind of see here, but it's kind of like gold, all these areas here. But you could think of it like what Primase does is just lays down. Again, you're going through the streets, right, to your house. You can think of it like there's plenty of potholes and intersections. So what Primase does is basically fills up those potholes with gravel. It's a temporary solution, not permanent though. So when Primase synthesizes a series of short RNA primers, but then it is replaced by DNA polymerase. So DNA polymerase, it's thankful for filling those potholes while it's traveling, let's say, to its origin, to its home, but it replaces those potholes of gravel with, let's say, powdered cement. As we know, powdered cement itself is strong, but powdered cement is not strong enough. So that's when DNA ligase comes in. Kind of, you can think of it like it adds the water and it joins the discontinuous fragments together, solidifies it. So this is primase. It synthesizes a series in blue of short RNA primers, or the gravel, and then DNA, and here in gray, is then replaced with it by DNA polymerase, and then it's glued, and here in highlighted, and basically joins the discontinuous fragments. Just extra information, the Okazaki fragments are usually around 1,000 to 2,000 nucleotides. In eukaryotic cells, they're usually around 100 to 200. And the potholes that primase fills with short RNA primers or RNA nucleotides, the gravel, it's usually around 20-ish nucleotides. Then once DNA polymerase comes in, it replaces it with DNA. DNA ligase definitely needs to come in because, again, DNA polymerase replaces the gravel with DNA, powdered cement, but DNA ligase, mixes it with water, joins the discontinuous fragments together. One more time, Helicase unwinds the DNA at its origin of replication. The second part is that Topoisomerase relieves the supercoiling. The leading strand, which is the new strand, is synthesized continuously by DNA polymerase. The lagging strand that's broken is the new strand in the bottom and is synthesized discontinuously or in parts. So primase synthesizes a series of short RNA primers or DNA nucleotides, which are then replaced with DNA by DNA polymerase. DNA ligase joins the discontinuous fragments together.